people who have the industry exposure. Like in KGI, there is one of the faculty who's worked with USFDA. So she comes and teaches the students, right? In Ramaya College also, there are people, one who worked in the industry in Sinjin and who is now left and is into teaching area. There's another professor also in that area. And if the faculties do not have, suppose they're being completely in the academic area, we bring them to Biocon. We spend 2 weeks training them on the industry part of it, showing them the industry, telling them what happens in the industry, and then they teach the students. So the focus here is that the education partner will give application-oriented teaching. They will constantly evaluate the students. They will see that the certification is given by these institutes. So if KGI is giving a certificate, it is a US-based certificate. So there is credit points associated to that also, right? So you get certificate from these properly governed education bodies, and they have proper evaluation systems. They have learning management systems. So they score you, you measure you at every stage. They ensure that there is projects to do, that there is case studies, that the students are interacting well. So that is the role of the education partner. So if you join Biotechnology, your morning first half goes with the education partner teaching in application oriented way. The second part, which is the functional visit part here. Right. So at this part is where you'll actually go into the industry. What happens here? The students go into the, if you take an example of Biocon or Syngene where they go and learn, they spend at least 2 weeks in each of the departments. Every afternoon, they go there and they see what is actually happening. So what I learned in the morning from the education partner, I come here and see how is it getting applied, right? Oh, this is the way they're using the biofermenter. You actually get to see the industrial biofermenter and the scientists who are working there in Biocon, who has many years of experience there, actually uses their time to teach these students. And it's not just one batch we are talking about. So that is also a part of the corporate social responsibility. Biocon is giving back to the society by ensuring that the scientists who are working there are always spending little time with the students, teaching them and passing on that knowledge that they have accumulated over these years. Right? So that is what happens in the functional visit. Now, this is the hands-on training part, right? In the hands-on training part, we are partnered with Narayan Hridayalaya, Thermo Fisher, and with Biotech, where depending on each course, right? Doesn't mean you join any course in Biocon Academy, you will go to all of them, or they all will be your education partners. These education partners and the hands-on training partners will depend on each course, right? So in functional visit, when you go there, you're not touching anything because it's a GMP area in Biocon. You're not employees of Biocon, you're students of Biocon. When you go there, you can't touch and do anything over there. So that is why we have done hands-on training with Biozine, Thermo Fisher, and Narayan to get hands-on experience. You do something with your hands. You see the techniques that are being, you see the instruments that are being used in Thermo Fisher. You see the fermenters that are being developed in Biozine. How do they all work? There you get to touch and do things over there. So that is also very important. So that is the third part of our model, which is the hands-on training. So recently I had read that if you have, 25% of the technical knowledge, it's enough. You can still learn. But 75% belongs to the soft skills. Now, what are soft skills? How do you communicate? How do you present yourself? Right? How do you, mix and work well within a team? How do you attend an interview? In an interview, you get only about 5 to 10 minutes to when the person makes a decision. Is that a measure of your knowledge? No. In that 10 minutes, how are you able to impress the person who's sitting opposite you with your knowledge, with your positive attitude, and with your communication skills? Everything matters. The way you dress, the way you speak, the language that you use, right? Everything matters. So that is the soft skill training part. And we do take care of that too, that to make the students who come in there to be more confident and ready to get into the industry. We talk about team building, we talk about presentation skills, we teach you time management. Because once you get into the industry, so many projects coming together to you at the same time. What do I do? How do I prioritize? Right? How do I manage all this? So there are tips and tricks to do that, and we teach you that. Even writing a proper email, it gives you a first impression. You're writing a mail to your boss, boss decides, oh, this guy doesn't even know how to write an email, right? So we teach you that too. So we have an external company who participates with us in that. We also have an internal communication coach. So the person, she works, the internal communication coach of Biocon Academy, ensures that daily there is touch points with the students. And every time if there are any weaker students who needs little more help in the communication, she personally spends time with them to ensure. So every student, we have finished 16 batches and we are now going to launch our 17th batch in the KGI with KGI in the biosciences area. I have seen every student have become definitely better within the 4 months. Though I've been in the field of education for 25 years, I always feel for a person to improve their soft skill, it requires more time, right? But here, I've seen this change happening, and that is because whatever you're learning, you get to apply it. If you're learning presentation skills, you have to do so many presentations throughout this course that constantly you're improving. I'm improving my body language, I'm improving my eye movements, I'm improving my speech. So I get to practice it, and practice makes a man perfect. That is why these students, once they finish the course in the academy, they are much more confident and can speak really well. This is the model which we apply to all our programs. I'm changing the slide. Okay. Now, pedagogy. What does pedagogy mean? It means the methods that you use for teaching, right? So, why are we so different? What is it that we use in our teaching? So, it is a proactive learning model. What does that mean? So when students, we normally when we go to any college or school, what do we tend to do? We just go and sit there. Then the teacher comes there and starts teaching. Some interested students really write down notes, focus. Some students are just sitting, "Oh, and then this go, I have to go and play this game now, I have to do that thing now, there's no focus, right? This is a proactive model where you have to come prepared, right? The class begins with a quiz. So they would have given you reading material which you need to read and come, and then there is a quiz. So that way you are coming prepared into a class, and then there is a discussion, right? You learn a lot from your peers also. It's not just the teacher who's teaching you. All the students who are there, they come with varied experience and varied degrees. They also come with knowledge. So peer-to-peer learning is also an important part of it. So you come prepared and you need to stay active in the class. There is marks for interactivity. If you ask questions, there is marks for that also, right? And there's also project-based learning. So throughout the course, we have a project which you keep contributing on. Every time you're reading a subject, you need to work on that project too. And at the end of the entire course, you're submitting your project. So every time you're learning, you're able to apply it onto a project. Case studies. Case studies is a very important tool because the industry brings in cases and says, okay, this is the area that we faced a problem. If you come across such a problem, how will you do it? And that makes us think, right? And that makes us contribute better and apply our knowledge better. So we do a lot of this. Most of our learning is very interactive and it is where you get to apply things. Just give me a minute. Okay. Okay. I think I pressed it too fast. I need everything to move fast. So. Okay, so now to tell you a little bit about the programs that we are doing in Biocon Academy. So in 2014, we had started it off with the Biocon KGI certificate program in biosciences, and we have finished 16 batches of that program. And the success is that 100% placement record across all 15 batches, and the batch 16 placement is still going on. right? So that's the important thing which attracts students to us because we are able to place all these students in different industries. So we have lots of companies coming to us to recruit these students. And why do they come to us and not go to colleges and recruit? Is because these students already have the industry skills. So the amount of time a company like DRL or Indus has to put on these students become less. It's not that they don't have to train these students at all because it's a 4 month certificate program, and there is much more to learn than that, right? But it becomes easier for them to teach because you already have some level of knowledge. Then we need to only train you into the next level. And in 2015, we launched the Applied Industrial Microbiology program with BITS Pilani. And here also the model remains the same. And the BITS faculty actually teach from different campuses. So they could be in Hyderabad, Pilani, Goa, and they teach from there. Then we launched one for the managers of Biocon, which is the Bioscience Management Program. And we are now trying to convert that to a full-fledged MBA program. That will be our biggest program because we are focusing only on small-term certificate program. But this will be for working people who have around 6 or 7 years of experience and want to move on to the next level and gain much more knowledge. We are planning to launch an MBA. Due to the COVID situation, we couldn't do it. Otherwise, by now, the MBA program would have started. Then we launch another program in the area of clinical development. So what we do is we look into what is the industry asking for, what are the skills that is required for the industry, what are they saying they're not getting in the market. Then we focus on that particular area and we develop a course in that area. For the faculties who have joined into this webinar, we also have a faculty development program, which is for 2 weeks duration. Naturally, you can't come for a 4 month course and so on, but this is a 2 week duration. We try to keep it somewhere around June, July when you have the vacation. You can come in and do this program. And here, all the scientists of Biocon actually come and teach you in the morning, and then take you into the industry and show you what is happening in the industry. This helps you to get an idea about the industry. It also helps you to guide your students better, to teach them better, to help them at least understand which department would they should be working for, right? So this is every year we have this program. Whereas the KGI program, we launch 3 batches in a year. But the faculty development program, the CDP program, and the AIM program, we do 1 batch in a year. If you go into the Biocon Academy website, you will see the dates of the when the batches are going to get launched. Then our latest program was in the area of quality control analytical with Ramaya College, and 1 batch is over, and we are ready to launch the next batch as soon as this COVID situation slightly settles. We are supposed to start in August. We might have to delay it a bit, but we definitely plan to launch that. The other areas that now we are looking into is global regulatory. And I also told you about the MBA in biotechnology. We are looking at integrated drug discovery for Syngene. Syngene is a clinical research organization. So we are looking for training the freshers in that area because Syngene wants to absorb those people. And Syngene is also looking for their current employees to go through this training. We are looking into a quality refresher program for Biocon junior employees, and we are also looking for a technical writing program for Syngene. So we help these Biocon group of companies also to train their employees in small ways. Like I said, Biocon is a CSR initiative. Biocon Academy is a CSR initiative of Biocon, and I said we place the students across different industries. These are a list of some of the industries that we have placed our students. We couldn't fit in more here, but every batch that we do, we increase. The numbers keep increasing of the companies who hire from us. You look into any of the top biopharma companies, they are over here. We also have startup biotech companies taking our students. And if you're interested in knowing which departments do these students get placed in, most of the students go into production, R&D, and if you take the whole of quality, I have split it into quality assurance, quality control, and quality control microbiology. But if you take those 3 areas, 64 plus 64 plus 54, so that is the biggest area. Quality is the biggest area where our students get placed, and then comes production and R&D, and then the other departments. So it's very interesting to see how the students want to get into different areas like corporate communication, project management. And that's also not bad, right? You don't have to be the technical expert alone, but if you have great communication skills and corporate communication is the area that you want to work in, having added biotechnology knowledge or a biopharma knowledge is always required when you get into such roles, right? So if I know chemistry, I have done my post graduation in chemistry, and then when we talk about any chemistry related courses, I will be able to pitch it. So that way, if I'm in HR or if I'm in the finance or if I am in medical writing, any of these areas, the knowledge of your industry definitely helps you to contribute much more. So our students go into those areas also. We've seen many people go into the marketing area too. They are interested in that area. So it always depends on what is your passion, what is your interest, right? Not what is happening over there. So, okay, R&D roles are there, let me join R&D. Oh, quality roles are there, let me join quality. So where is your strength lying? What is it that you want to do? They say if you have the passion, you never feel that you're getting into work. If you have the passion, your Monday mornings and your Friday evenings are the same. You're excited about it anyways, right? They always say Monday morning blues and Friday evening people are generally happy. But if you're really passionate about your job, it doesn't matter. You will love to come into work and contribute so much. So passion matters. And this is where you can find our details. You, if you have any queries, you could call us at that number. And we are located in the Electronic City Phase 2. In our Biocon Academy website, we have all the details of all the programs, what are the content being taught there, and who are the faculties of these programs. All details you'll find, plus the fees. I'm sure you're thinking about the fees also. The fees varies for different programs. So if it is a AGI program, it's pretty expensive because it is a US based program. And if it is an Indian program, then the fees is lesser. So you can look at our program rates and everything. It's available on the website. I want you to leave you with a few thoughts. It's not enough, like I said, if you just have the degrees and the education. A few things that the companies are now looking for, and the foremost one is innovation, right? So if you keep doing the same thing, you're not going to survive for a long time. You should be ready to be flexible. You should always innovate. You should think what can be done. There are a lot of startup companies which have come in, or big companies. If you can learn their history and how did they start, even when you're taking at the KFC or the Madurai Eye Hospital, how did they all start, right? You don't need to have lots of money and lots of knowledge just to do something small. You can start in a small way. If you have a great idea and you think it's going to work out. If you see the Royal Enfield bullet that you have, you go to Punjab, they actually converted into kind of a tractor that can be used, right? So people are really innovative. And in Punjab, they also use the washing machines to churn the lassi because lassi is something which everybody drinks there and you need loads. So they use the washing machine which goes in speed and makes the lassi. That's innovation. People, all of you have are born with that. So focus on thinking more and getting your ideas out there and seeing what else can you do. Especially in this current COVID situation, when different markets are not able to cope up, you need to innovate. You can't just say, I am in this, now my job is gone, what do I do? I just sit and wait for the situation to get better? No, think what is required now. Which companies are doing better now? Zoom, right? Zoom suddenly picked up so much. The person who owns Zoom has become multimillionaire now, just with this space, right? And if this time you're launching some tool that is useful for these, like the WebEx that we're doing, if it is useful for these technology teaching online, it will be a big seller, right? So if you're in the IT area and your job is getting impacted, you could think of developing something like this. There are so many small, small things that you can do and keep changing, keep learning, right? So innovation, learning, and networking. It's very important, very, very important that you connect with people. Say, suppose one of my team members wants to go to US and do some project there, or learn something there, and she's not got an opportunity to go there. But she's told me many times that if I get an opportunity to go to US, it would have been so wonderful. And she's told many of her friends too. She's told many of her dad's friends too. She's told everybody that she really wants to go to US and do this. So what happens is that if someone comes to me, one of my friends comes to me and says, there's an opportunity which has come up in US. You're thinking of hiring somebody. Do you know of someone? That'll strike me. Oh yes, my team member had told me about it, and I'll try and connect that. So networking is important. Stay in touch with the right kind of people. Have mentors, right? Look at the person who can really help you and always seek guidance from them. Always be in touch with them. The people who have so many years of experience can be real good mentors for you because they have struggled and reached there, right? It's not been an easy way for anyone. So they have gone through so much, and that experience you will not get anywhere in any of the books or in the Google sites. So you need to have mentors. You need to listen to their feedback. You need to be open in networking, not just be on Facebooks and Instagram, be in LinkedIn also. You have to become a professional. Right? So now I look at if you have any questions, I could look into the chat for any questions.
Thank you very much, madam, for a nice presentation.
Thank you.
Participants have some two questions, madam.
Okay.
One is, since students cannot develop their hands-on skills labs in the labs right now in COVID situation, what kind of online courses would you suggest which would help in industry labs?
Okay. So this COVID situation is a pretty bad situation as we are not able to launch our courses also. But the methods that we are trying is like if you take Biozine, for example, they are into biofermenter training. They will also be starting their programs with lesser number of people. So it's if you still take all the precautions of wearing your mask and sanitizing. Uh,
And one, madam, thank you very
one,
much. And
one second, Umesh, I'm not finished it yet.
oh, sorry.
No problem. So there are companies which are running these programs still, and there are a lot of online programs like
as told in the morning. So here you could see briefly, I'll take you. So I am not going to tell all those micro, biochemistry, food technology, genetics, environment, plant, animal, IPR, bioinformatics. And you could see biotechnology in India in 1978, first biotech company, Biocon. And we are lucky that we are with Biocon Academy. And you could see that Kiran Majumdar Shaw madam started with the company in 1999. Now it has one of the major bio industry in the world. Then in 1981, we have CCMB in Hyderabad. Then Lalji Singh, who pioneered the Indian DNA fingerprinting. Then in 1986, Department of Biotechnology. Why I am emphasizing this, 1986 Department of Biotechnology. So we should be grateful, blessed enough that government of India, not only India, pan world, they have a separate unit called Department of Biotechnology. In Karnataka, we have IT and BT. So therefore, we shouldn't be, no biotech this, biotech that. We should be very focused. And since then, gradual, regular process has been made the field, and we should be technically sound to uphold the purpose of the biotech inception. And what is the significance? As you could see, biotech industry has grown threefold in last 5 years. Industry has increased spending in R&D and attracts FDI, drugs, pharma, biotech industry is ranked third in Asia Pacific region. After Japan and Korea, there is budgetary allocation and improvement in the skilled manpower infrastructure and contribution to the improvement of agricultural sector. Now you could see our world is reeling with corona endemic or pandemic, what we are seeing it. What we are dependent on? The drugs. If drugs comes from the biotech industry or the pharma industry, I think it makes a multi-billion or trillion dollar, the business. So therefore, you could see not only it helps support the agri and human or health, it also supports the life. So humanity on the top priority. Then we have the BIREX, then DBT, then biotech industry partnership programs, introduction of biotech bill in the parliament and setting up the regulatory authority to oversee the progress and so many things to carry out the risk assessment of all the biotech products, because we have to look even the ethical parts also. Then the issue areas, as you could see, multiple regulatory bodies, funding constraints, lack of infrastructure. So to overcome this, what we have to think of a joint venture where all of us joined together in a single platform. Not like that, a small organization and doing individually and don't land up. Instead of, we can think of multidisciplinary, interdisciplinary approach to overcome all those. So if you see the great organizations like Japan and all, if you could see, no, I'll just quote a simple example from Hebbal till Yelanka, that entire set is one set of industry. So one process, one thing, then another goes like that, no, and it is a collective effort. So we should think in that way. Then Biotech at PU Chandigarh and all those things, so you could see DBT, DST, UGC, ICMR, CSIR, and the current courses. What is most important for the present generation, they should understand that normal this BSc courses or MSc courses or BTech courses, or no, if you forget anything, just a BSc and MSc will land up or make you land up in a highly paid salaried jobs. So for that, briefly, I'll take you a few slides, teaching, research, pharma, food processing, bioprocess, agriculture, setting up own project. So this is entrepreneurship skill you develop with. So these are the job opportunities, no, which I'll take you, like applications are in medical, therapeutics, agriculture, environment, animal, industry. So you could see these are the multiple things where you can look into. So now, as briefly for a few minutes, I'll take you to the part which I'd like to really focus on is, I think, yeah. So you could see here a thought to start with, Charles Darwin, The Origin of Species, and you know that we are looking into that survival of the fittest. Our eminent industrialist Ratan Tata has clearly told, this year we are not supposed to look into profit and loss. What we are supposed to look into? Survival of the fittest. So therefore, it is not the strongest of the species that survives, not the most intelligent, but the one most responsive to change. So change, we should be flexible, we shouldn't be rigid. And our Indian tradition has clearly mentioned how flexible we were. There are pros and cons that for flexibility, but we shouldn't forget our roots. So Charles Darwin, The Origin of Species. So can you sense the change here? So what will take you is the emerging world is going through a major change, either through environment, through lifestyle, through health, everything else, resulting in a borderless world where 7 I's are free flowing. What are the 7 I's? Investment, industries, information, ideas, innovations, and individuals. And what is the 7 point? These individuals shouldn't think of individual gain. They should think of a collective way that is called compassionate, empathetic, and humbleness. And you could see the types of academic courses which India runs with. So these are the job-based course where you could see, and as we are speaking about biotechnology, and I being into this sector for more than 20 years, I could tell you that many of my friends after doing biotechnology research and all, they have joined into IT companies and other companies or shifted. But I personally tell you that, I quote my own example, stick to your field, you will success one day. It's like a mango plant. You plant for a day. You can't expect immediately within 6 months or 1 year. You have to wait for 5 years to get the nice dividends. So here you could see we are telling about research-based courses. So biotech comes under this. Research-based courses where you have BSc, MSc, BTech, and all those life sciences, and immediate jobs are very less, that you should understand clearly. PhD or minimum PG is compulsory for better career scope. So I'd like to focus here that immediate job after 3-4 years in other courses, but here you have to minimum do PG and pursue PhD. Why? It requires highly rated ranked premier institute for better job opportunities, and Biocon want to be one of the academy which supports Wipro, Biocon, and so forth, the BITS, Pilani, IITs, everywhere you could see IISERs are there. They have their biotech units. So for better job opportunities, since it requires highly sophisticated lab facilities and highly qualified faculties. So therefore, biotech is not like that overnight you can open and you can run the show. And you could see this is the hierarchy. So affiliated college, private, autonomous, state government, central universities, JNU conducts all over India biotech courses, BHU, Punjab University, central universities, premier institutes are there, as I have told you, IITs, NIT and IIS, and research councils. So we have to reach here, the second in the rank or the second pyramid, second from the top. Because once we land up here, then we can think of going ahead. And these are the organizations which help you in your studies for either JRF or SRF and to do hardcore research. So these are the educational institute. From there, you have to raise your bar or raise your level to research concepts. And this is what individual autonomous research organizations, IIC, TFIR, NCBS, and JSIR, and all we should be proud enough that they are in Bangalore. So hierarchy continued. So here you could see, as we go in this hierarchy, education is free, funded, get monthly stipend with better facilities and career scope, which I'd like to emphasize that you should think of stipend and pursuing your higher studies, and lots of stipends are there, which runs more than 25,000 per month, which by just doing your 3 years course and running into the job, which Bangalore, many of the organizations, KPO, BPO, and all those sectors, I will suggest you that as you are biotech students, please focus on those fields. As we work hard, we get better institutions, hence facilities and faculties, and thus better opportunities, and we decide our future. So jobs in biotech, India needs more than 30 lakh PhDs by 2025 and more than 1.5 lakh research scholars as we open up our economy and aspire for superpower status. And why superpower? It's going very apt. If you could see now 2020, we are now, just few years back, if we see, India was never considered to be a powerful country. Now you could see US, Russia, France, everyone is supporting not only by defense into the terms of research and all those. And for that, what we should do, we should pursue quality PhD research scholars who are emphasizing to empower India indigenously and going with our Prime Minister stating that indigenous, make in India. Though PhD is the minimum decent qualification in biology, if you plan for your long-term profession, still there are limited growing opportunities. As I had been clearly telling you, I give my option, or I give my a simple motto to all my dear students and to in fact all the participants: 5 years versus 50 years. 5 years what? 3 years of UG, 2 years of PG, and after that you write a decent entrance exam and get into the scholarship stipend, and your life is settled for 50 years. And if you think of enjoying those initial 5 years, then I don't have to say anything else. You can understand. So what are they? Let's explore them. So these are the, no, I need not, it is a self-explanatory. You could see who tells that biotech doesn't have government sector jobs. You have private sector, R&D, manufacturing, marketing, and second, you could see government sector jobs, non-fellowship fresher, and fellowship-based academic jobs, higher education, research, and non-core and other jobs. So private industries, as you could see, all pharma, BT companies, Biocon, Dr. Reddy's, huge investment in chemical instruments and other lab, lesser manpower are required. So you require skilled. A simple, again, I'll tell you, one of the student who just pursued MSc in bioinstrumentation and runs a premier electron microscope center in Jalma, that is ICMR Institute. And you'll be wondering to know his salary, it's more than 2 lakhs. Because even the expert he has undergone, he is like, the electron man is been told in ICMR Jalma, that is in Harvard. Prefers premier central top-rated private institute for fresher recruitments. So who tells that freshers are not being recruited? Only thing is that, as Biocon Academy Bindu Madam has clearly told, you should have feather on your caps. Not like just you are doing UG coming to college like a general other course. Biotech, you should understand, it's a professional technical course. You should keep adding feather to your cap. So better job facilities and better skilled candidates. And small companies, medium, big, and multinational companies are there. And we should be proud that India has got one of the international unit, if you remember, that is called ICGB, International Center for Biotechnology, which is in Delhi. One is in South Africa and is one in Italy. And these are the private sector. So no, this is self. I need not to keep on explaining you. Only I am telling about the skill need, limited, moderate, technical, R&D, huge investment in lab infrastructure. So you could see experience of 2 campus placement or backdoor entry, which is reference which you have to omit completely, because always it should be your first choice for most job seekers should be your enhanced skill, professional skills. Then the first focus should be on the top, no, bottom. So bottom is marketing, manufacturing and R&D. Now you decide where you want to be. You want to be in marketing, you want to be in manufacturing, you want to be R&D. And I see that most of my students, after their MSc and research, they have landed either in Milan lab or in, if I am not wrong, Teva Pharmaceuticals, Bharat Biotech. They are all just by doing BSc Biotech, MSc Biotech with enhanced skill, they are there. So you should understand that private sector educational system, and so you could see that government sector jobs, yes, which briefly I would like to tell you. Research-based job does not mean PhD, but it may or may not accompany PhD. So research-based, as I have told you, one of the scientists in Jalma, he has, after doing MSc, he is into electron microscope center. So there are fellowships, which I was about to show you. You could see fellowship-based fresher research programs, CSIR, ICMR, ICAR, GATE, first class pass in science or engineering after MSc. So if what you could see, less competition, merit is honored. So highest pay scale initially, 16,000, 19,000 of fresher 20 to 24. I tell you that just after doing other courses, who will give you after 5 years this? So therefore, we have to blend, as no, there was a talk that MBA biotechnology, yes, because that is required, because we should understand a commerce MBA graduate can't lead a biopharma industry because their technical part will be of commerce background. We should think of R&D, as I have shown in my earlier slide. First is marketing, manufacturing, and then top R&D. So the government, more opportunity in central and labs and all those, and you could see we get circulate minimum 300 fresher research jobs every month across India, only from government sector at biotechnologies2020.com. So you can go through this website, which will help you to understand that how much you stand being a biotechnologist. And as you could see here, academic jobs, PhD eligibility, you could see here 10 plus 2 plus 3 plus 2. So the total you spend around 12 plus 13, 14, 15, 16, 17 years. Then these are the GATE, fellowships, stipends, you could see here, and sample in number, all India ranking, you could see CSIR, UGC, NET. So where you are landing, CSIR labs. So academic teaching jobs, lectures with NET are eligible for a limited stipend, whereas, as I have shown you in my earlier slide, with PhD or into that after BSc and MSc, you land up with 24, 25,000 under different scheme, ICMR, then all those programs. So higher education and research, as biology is predominantly a research subject, having a PhD or minimum a PhD is must for better scope. And I just complete with this, you could see these are just wanted to show you that more than 70% BTech student like basic sciences like genetics, molecular, and all those are better job. And this is the call for the day or call for the time that during this COVID-19 has made us learn that how important our life science courses are. So with this, I just conclude that my talk stating that this biotech is far more better, but as Bindu Madam has clearly stated that short training and certificate programs will be the feather on your cap. Thank you so much.
Thank you, Dr. Shantanu, for making a wonderful talk within 10 minutes, and you gave a very good overview on scope for biotech research and also job opportunities. Thank you very much. Now I request Dr. Ramya Madam to give a talk on opportunities for research. Madam is basically a biotechnology expert. She has done a PhD in biotechnology. we have two dynamic principals for today's webinar, Dr. Shakuntala Samuelson and Dr. Ramya Madam, whom I know for more than 10 years. We have organized many programs in association with them. Today, Ramya Madam will be giving a talk on opportunities for research in biotechnology. Over to you, Madam.
Thank you, sir. A warm welcome, Dr. M. Ramesh, Chief Executive Officer, KSTA, Dr. S.S. Ishwaran, Academic Dean, Biocon Academy, Bengaluru. Thank you so much for coming up with such an amazing platform, webinar, a need of an hour for our students as of now. Dr. S.N. Venkatesh, Principal, Seshadripuram First Grade College, Professor Shantanu, Secretary, North Bangalore Science Forum and Head Department of Biotechnology, Seshadripuram College, Dr. Shakuntala Samuelson, Principal, Surana College, South End, and Sri B.G. Umesh, Scientific Officer, KSTA, for coordinating such a lovely event. And I am so very happy to look at the participation number. That's 175. So I am very glad that there are so many students out there with all this pandemic and uncertainty of what's next. They have been enrolled for this platform and looking forward that probably there is a few of us who are out there for them, probably guiding us through this particular pandemic situation and hopefully seeing to it that their futures are being well secured. Thank you so much. Just give me a minute. I will just share my content. I hope I'm audible.
Yes, madam. Yes, madam.
Thank you. Just to give a minute, sir. I'm not able to connect to my desktop.
Yes, ma'am. Now connected.
Yeah, just give me a minute, sir.
Give a brief overview of his expertise. Ankur is working as General Manager, Process Development at Biocon R&D. He has more than 18 years of experience in developing and scaling up processes using mammalian and microbial platform. Ankur has extensive experience in leading teams, setting labs and pilot plants, technology transfer, and was integral member of teams involved in getting important product approvals for Biocon in various regions like US and Europe, etc. Ankur has masters in biochemical engineering and biotechnology from the most prestigious IIT Delhi, and he has an executive MBA from another great institute, IIM Bangalore. He has served as advisory board member of School of Biological Sciences, IIT Delhi. Ankur has won Great Manager Award conducted by Economic Times and People Business in 2017 in the overall management category across industry. His introduction, let me welcome Dr. Ankur Bhatnagar for this webinar. He will be delivering a talk on an overview of development of a biological drug. Over to you, Dr. Ankur.
Thank you, sir, for the kind introduction. Am I audible?
Yeah, please.
Okay,
Yes, sir.
I will try to share the screen. Screen also visible. Is the presentation visible?
Yes, sir. Yes, sir. Very
Okay,
much, sir.
thank you. So I would like to start by thanking all the dignitaries for this opportunity and very nice talks. They were very informative. I have been listening to all the talks till now, and want to thank you for all the audience. And so the topic for my presentation is an overview of development for biologics. So just setting the expectation for the talk. So, developing a biological drug generally takes between, 8, 10 years to 20 years. And we have around less than 1 hour to be able to cover the process for developing a biologics drug. So what we will do today is try to have an overview, try to see, most of the important points which, have to be, paid attention to while developing a drug. And, I think you will realize that what we were hearing in the previous talks, different skill set is required. And throughout the presentation, you will realize how different skill set are required to be able to make a drug from the discovery and to the market. I will start by saying that drug development is generally, there are two main arms for it. One arm is called as the CMC. The full form is chemistry, manufacturing, and control. So the CMC arm is actually about making the product. So it's about identifying the molecule, discovering the molecule, making a process to make that drug. How do we scale up to a manufacturing scale? How do we make sure that we can consistently make the drug, batch after batch? We have to get approval from the regulatory agencies, and then we get inspections, we get audits. So all that is the part of CMC. The other parallel track which goes on, along with this CMC development is the clinical. So the different stages of clinical is that the drug is tested in animals first to find its toxicity, then in some patients, first a limited number of patients where we study the amount of dosage which is required, then the set of patients become more. So we have phase 1, phase 2, phase 3, and all this data from the clinical side and from the CMC side is submitted as a final package, which leads to an approval for a drug. So for today's presentation, we will be focusing on the CMC part. That is where the process development comes in. So these are the high level, steps in developing a biologics drug. So first step is identify the product. Like in any industry, you have to first find out the product that you're trying to make. And then once you identify the product, you have to find a good machine which can be used to make that product. Find a machine, you have to develop an entire process to make the product. Then the process is first made in the lab scale or in the pilot scale, and then finally this process has to go into a manufacturing stage. Then you have to develop a lot of testing tools to monitor whether the product that you're making is right or not. Then you have to establish a shelf life for this product, which is important for our drugs. So all the drugs have certain, expiry. And then finally, all this goes into, a package where we get approval for making this product for the patients. So I will try to cover all these, steps. The first step is identify the product. So this slide talks about and gives you an idea about the complexity which is along with these biologics molecules. So if you see on the left hand side of this graph is aspirin, which is a chemical molecule. And then we have proteins like insulin, erythropoietin, and then finally monoclonal antibodies. And the numbers that you see are actually their approximate molecular weight in Daltons. So you can see that, the complexity, just because the size of the molecules is much bigger, goes higher as we go to more and more complicated biologics molecules. So today, what I will be focusing on is more of a monoclonal antibody as a product. So just to again show some comparison between the other pharmaceutical, which is more chemistry based, and the biologics. So the biggest difference is in the size. The other big difference is that they are made using chemical synthesis, and all the biologic products are used making the living cells. Usually, the small molecules pharmaceutical products are given orally, but these biologics cannot sustain the harsh environment of the stomach, so they have to be injected or they have to be infused. The small molecules you can get through retail pharmacies, but these complicated biologics are only available with the doctors or with the hospitals. So there is an example, below, which is a Lipitor and Herceptin as a molecule. So while choosing the product, and as we decided to have monoclonal antibody as a product, so monoclonal antibody can work by various mechanisms. And as we identify the disease, we have to identify the mechanism by which this disease works, and then identify a molecule which can help us in blocking that mechanism. So on the slide, you can see in the orange blocks, they are all different mechanisms by which a monoclonal antibody can actually work against a disease. It can be blocking, so these antibodies can go and block, and that is how they prevent certain reaction inside the body to happen. So this is some example for that is for people who have studied immunology, say anti-TNF alpha. There are a lot of cytokines which actually play a very important role in the autoimmune diseases. So if you have autoimmune diseases, and if you want to block the chain reaction for the autoimmune diseases, you can have an antibody which can go and block some of these cytokines. So that is how it works, and it slows down the progression of the disease. It can be through ADCC. This is antibody dependent cell mediated cytotoxicity. It can be by targeting. So the antibodies are known to be very specific for their targets. So we can attach a toxin, or we can attach a toxic molecule to that antibody, and the antibody makes sure that it goes to the right target and binds to the right target. So this helps in reducing the side effects which are generally associated with other toxic therapies like chemotherapy. So the idea of this slide is that once you have decided the disease that you want to work on, and then by understanding the mechanism, we have to develop a molecule which will prevent the progression of that disease. So this is a little more detail about the structure of an antibody. The left hand side, like all proteins have form and function, so they have some structure and they have some function which is attached to that, related to that function. So on the left hand side of the diagram, you can see the biological characteristics. So this is the biological characteristics of an antibody, which defines how does this antibody work. So there's an antigen binding site, there is an effector function site, which is the FC portion of the antibody. So this helps in the ADCC mechanism for the antibody to work. The antigen binding site makes sure that the antibody goes and binds directly to the right spot where we want it to bind. So these are the biological characteristics. On the right hand side, you see the structure of the molecule. So this is, like all proteins, this is made up of amino acids. So we can have a lot of heterogeneity in that. There are places where disulfide bonds are formed. There are places where glycosylation can happen. There can be some fragmentation which can happen, like at the hinge region, one light chain can get cut off, one heavy chain can get cut off. There can be some aggregation which can happen. So there's some antibodies, molecules which can aggregate. So all these are the different physiochemical properties of an antibody. So together, like any protein, the biological characteristics and the physiochemical characteristics play a very important role in making sure how does that antibody work and how safe is that antibody. So once we have identified, the product that we want to make, the next thing is we have to choose the machine to make the product. And the interesting thing for these biologics is that scientists have not been able to make machines which can make these complicated biologics molecules. So we have to use some living cells to be able to make these products. So we'll see the examples in the next slide. So some examples are we can use bacteria to make certain products. So generally more simpler products, proteins like streptokinase, we use bacteria. We can use yeast. So yeast can be used to make a little more complicated product. So we use yeast to make insulins, for example. And then the last is mammalian cells. So the cells which are very similar to what we have in our body. And we need mammalian cells kind of cells as machines to be able to make these complicated monoclonal antibodies. So again, a reason why we have different machines for different products. So the best glycosylation which can happen, which is very similar to the body, our body, comes from the mammalian cells. The bacterial cells cannot do glycosylation. Yeast cells, they give a hyper mannose, which can be immunogenic to patients. The insects and the plants also have a different type of glycosylation. So the best fit to make a monoclonal antibody is using the mammalian cells. So a lot of textbooks about procedures or the methodology to make monoclonal antibodies, which is like we immunize a rat or a mouse, and then we use that to create monoclonal antibody. So this is not the way we make it at an industrial scale. This is still used for working on a smaller scale, but for industrial scale, now we use the recombinant DNA technology. So we have certain mammalian cell lines, and then we insert the DNA of the molecule that we want, or sorry, the gene of the molecule that we want to make. And once we do that, we get several clones which will have the gene inserted, and from that we have to do and select the best clone. And once we get the best clone, which has the right gene inserted at the right place, we have to preserve this because this is the most important part for making a product. So we freeze it, and we call it as a master cell bank, as a working cell bank, or a research cell bank. For mammalian cells, there are many cell lines which are available, which are commonly used. The most commonly used is a CHO cell line, which comes from the Chinese hamster ovary cells. So 70-80% of the industrial antibody products are made using CHO cell line. There are other cell lines also which are NS0, SP2O cell lines, which come from a murine origin. They are also used for many antibodies. Few other cell lines like baby hamster kidney cell line, Vero cell line, and there have been some human cell lines also which are available, but they are not very widely used because the risk of human virus coming from those cell lines. So, like I said, most of the industrial processes use CHO cell line. So we have discussed how do we identify the product. We have discussed after the product, how do we identify the machine. And then we come to the next stages. Once we have the product and we have these living cells, we have to develop a process to make the product. So the process to make these products at industrial scale, there are three main process steps. The first step is called upstream, where we use a bioreactor. So this is the stage where we actually use the living cells to make the product inside the bioreactor. The second stage is called downstream, which is the purification stage. So once the product is made, the job for the cells is done. We have to remove the cells. And then when the cells are there, they will also make a lot of other things. They will make their own proteins. They will have their own DNA. Some cells will die. So they will release their intracellular material. So there will be a lot of other things also along with the product. So the second step is the purification step, where we use different chromatography and filtration to be able to purify the product. The third stage is the formulation stage. So this is the stage where we get the purified product, and then we have to make sure this purified product converts into a product, a drug product which can be given in a market, which can have a shelf life for an X amount of years. So these are the three process steps which get repeated each time when we have to make the product. Upstream, make the product. Downstream, purify the product. Formulation, formulate the drug product. All these steps are going on. There are a lot of quality controls in between. There are quality assurance teams. There are manufacturing teams, so safety teams, maintenance teams. All of them get involved in making sure that these processes run smoothly. So before we go into a little more detail about each of these process steps, I just wanted to give an idea about the product that we are making are not simple products. They are highly complicated, and like I showed in one of the earlier slide, that they have both a form and a function. So they can have different masses, they can have aggregates, fragments, different type of charge, because there are a lot of charged amino acids. They can have differences in glycosylation. So you need tools to be able to monitor each and every product quality attribute. And we have to have a very tight control on each and every product quality attribute for the product that we are making. So during the whole development, during the whole manufacturing, we have to keep on building testing of the product, especially in the development, to make sure that we are making a process to make a good product which is safe and which is efficacious. So this this slide shows certain quality attributes that we use. We measure charge, we measure differences in size, fragments, aggregates, and all the protein analysis techniques that you all would be reading in your syllabus. We actually use them. HPLCs, mass specs, capillary electrophoresis, everything, every technology is used to be able to monitor some or the other product quality attribute. And generally, not one technique is used. We always have two or three orthogonal techniques. So if we are measuring size with one, we also have another orthogonal technique to be able to verify the charge distribution that we have. So we have circular dichroism. So all these are different techniques which are used. So we go to the next slide. So like I said, different techniques. So we have to understand the primary structure. We have to understand the secondary structure, and you can see on the third row, the third column of the table are different techniques which we all read in our curriculum, in our academics. All of them are practically used to understand from primary to the tertiary to quaternary structure of these antibodies. I also mentioned earlier that we also have to monitor the effector function, the functionality of the antibody, how strongly it is binding. Is it binding to the right target? Is it, is it creating the effect which it is supposed to do? And this can be done through many functions. And you can see we have to study a lot of bindings to different receptors of an antibody, and then compare it with some standard and find out whether the behavior of this antibody. All this is done in vitro. We have to develop assays to in vitro keep a check of the performance of the antibody throughout the development and throughout the manufacturing. So now this is a process flow which generally typically gets used, and you can see on the top side are the bioreactors. So we start with a vial where the cells are frozen, they are kept frozen, and whenever we want to make a product, we take out these frozen vials. Then we have to expand the cells because we have to run a large bioreactor, so you need a lot of cells to be able to inoculate a large bioreactor. So we have a scale up stage for all these cells. Once we reach the main production bioreactor, then we run the process there. Once the process is over, we go to the different steps of downstream, which are generally more on the second row, and then finally in the end we do the filling of the product. So I will explain each of this process step in little more detail. So but finally, if you just see in this one slide, we are starting with the vial, which is a vial of the living cells which have the gene to make the product, and the slide is ending with a vial which is actually the product which goes to the market. So it shows a vial finally getting converted into a vial for product. So this is a little more detail of the upstream process. So we start with the vial, like I said. We have to expand it to the shake flasks. Then we have to expand it to the seed or an inoculum bioreactors. And then finally we go to the main production bioreactor, or the manufacturing bioreactor, as we say. So in the next slide, I will give you some idea about what different parameters do we actually have to optimize when we are developing an upstream process. So generally, if you think the development of an upstream process is actually of two parts. So one part focuses on the optimization of the media and the feed. So the media that we use to grow these cells plays a very important role on how the cells will behave, how will they grow, how much product will they make, and more importantly, what will be the quality of the product. So left-hand side shows the media and the feed, and you can see nowadays we generally try to use a pure chemically defined media. So if you're giving a chemically defined media, then we have to make sure that each and every essential component is added. So we have to give amino acids, vitamins, trace salts. We have to give lipids. We have to give some, bulk salts like sodium chloride, calcium chloride. So all these have to be provided in the right amount to make this media. There are a lot of complications in making this media because, as written on the slide, this media has close to 80 to 90 components, and each of this component has a varying degree of solubility. So we have glucose and sodium chloride, which are very easily soluble, but we have certain lipids, we have certain vitamins, which are very difficult to dissolve even in small quantities. We cannot give very different we cannot use extremes of temperature, extremes of pH to make these medias to dissolve these components, because this will then the cells will not be able to grow under these extreme conditions. So all these things have to be taken into account while we are making the media, and we are making sure that we make the right media each time for the cells to grow. On the other side of the slide, you see the conditions of the bioreactor. So, like, two things are important: the media and the feeds that you provide, and the environment that you provide to the cells. And the environment comes from the bioreactor. So, for example, what is the temperature set point? that we are trying to control, what is the pH we want to control, how much share we are giving to these cells, and especially these mammalian cells are more sensitive to share because they don't have a cell wall, so we have to optimize the share also properly. What levels of waste metabolites the cells can tolerate. So, whenever we give nutrients to the cells use the nutrients, but they make the waste metabolites. So, the concentration of the waste metabolites are also very important. What is the amount of nutrients that we have to give? And the real, exciting thing is that a very small amount of nutrients, like I mentioned, small amount of magnesium, calcium, manganese, iron, they play a very, very important role. And, they can make big differences in the quality of the protein. And the reason is because many of these small trace elements actually are cofactors to a lot of enzymes in the glycosylation pathway. So if their concentrations vary, or you can try to vary the concentration yourself to get the right glycosylation that you want. So it is very exciting to study about the interaction and the behavior of each of these small nutrients. So to optimize the upstream process, generally optimize the media right, get the feeds right, and get the environment of the bioreactor right. So these are all the two areas in which we optimize the upstream. Coming to downstream, so what happens when the bioreactor or the fermenter run gets over? Like the diagram shows, you have the product, which is the antibody inside, but you also have a lot of cells. There are living cells, there are dead cells, there are cells which have completely lysed. There are a lot of intracellular particles which have come out. There are some nutrients which are still lying there unutilized. There are some waste metabolites which the cells have made. So a combination of all this is called the harvest, the cell culture harvest. And now the purification steps have to start. So the first step on purification is to remove the cells. So either we can centrifuge and remove the cells at the large scale, or we can use filtration to remove these cells. So once we have removed these cells, the purification of the product starts. The first stage in typically in a monoclonal antibody production is affinity chromatography. So we use the affinity chromatography and take out and try to bind the antibody to a ligand called protein A. So and then elute the antibody out. After that, there are multiple chromatography steps. We use cation exchange chromatography, anion exchange chromatography. In some cases, we use hydrophobic chromatography. So all these stages are actually used to polish the product. And by polishing, I mean removing trace amounts of host cell protein, trace amounts of the DNA which comes from the host cells, any endotoxin which can come during the process and can be harmful for the product. So all these are the polishing steps of different chromatographies which we have to do to be able to purify the product. Now, once all this is done, and then the final stage is the filtration of virus, and this filtration is different from the normal filtration because this is a nano pore size, whereas all other sterile filtration are for micro size. So this is almost the final stage for the product, where we make sure that all, if any virus is there, it is removed. And then after that, some stages we have to concentrate the product. So we make the product more concentrated. So if we have to do that, we have a filtration and ultrafiltration step in between. And after the product gets concentrated, we take it to the filling line. So this is all the downstream unit operations which are typically used in the monoclonal antibody process. And in the next slide, I will show you, again in a table form, what does each process step does. So if you see the attribute list, the first column says, the host cell protein, the host cell DNA, both of them come from the cells, the dead cells. Any virus which can come, the charged species, like I said, antibodies can form aggregates. These are proteins. They can form fragments. So all these, and on the second column where you see cell culture, that is the bioreactor step. So all these things are formed in the bioreactor, and then wherever you see a green, it shows in different purification or the downstream unit operations, these get removed. Like for example, since virus can be a big risk, a lot of safety is built in to take care of viruses. So if you see the virus step, so there are 4 places where we make sure that the virus load is reduced during the process. So this is what, and each step has to be optimized for the best condition so that it can give the right yield and it can get the right product quality that we are aiming for. So this happens in the downstream process development stage. Now, like I said, the third process stage is the formulation and the drug product development stage. So here there are 3 important parts to it. So first is we have to identify a formulation, and the formulation will be the one which will make sure that this purified protein is stable for the whole shelf life of the product. And what we mean by stability is that the quality attributes, like I mentioned, a lot of quality attributes, are part of characterizing a molecule. So we have to make sure that for the whole shelf life, if it is 2 years or 3 years, all these product quality attributes are actually maintained and they are stable. So we have to find the right formulation which will be able to do that. This is an example of a formulation. You can have sodium chloride, glycine, sodium hydroxide, water for injection, and whatever we add in the formulation apart from the drug product is called as excipient. The second important part is once you know the formulation, you have to develop a process which will convert this purified product into a drug product. So like we have upstream process, downstream process, you also need to have a drug product process to be able to make sure that you make the product in the right way. The third step is that once you have the right product, how will you make it, safe and convenient for the patient? So the product goes in different forms to the patients, and the later slides I will give some idea on that. So these are the 3 main areas for product development. Find the right formulation which will make it stabilize, find a right process so that you can make the right product, and once you have the product, make a right container closure for the patient so that it is safe and convenient for them. In the next slide, a little more detail about the formulation development. So like I said, everything which is added to a formulation which is not apart from the drug product is called as excipient. And what we have to optimize is we have to optimize the buffers to be able to get the right pH. So these antibodies have different PIs. We have to understand the PIs of these antibodies and then design a formulation according to that. We have to maintain an isotonicity so that the osmotic pressure of the drug when it is delivered is similar to what we have in our body. We need to have stabilizers so that during the shelf life of 2 to 3 years, the product is retained, and these are proteins. So you can imagine if it is a high concentration protein which is lying in a vial, there can be tendencies that these proteins can aggregate. Their hydrophobic, hydrophilic interactions can happen, and these proteins can aggregate or these proteins can fragment. So these stabilizers help to maintain an environment inside so that these antibodies are actually not aggregated or fragmented. We need to have preservatives at time. So because sometimes the drug is delivered as multi doses. So once it is opened, the preservative takes care that the drug doesn't get spoiled. There are surfactants, so which reduce the surface tension, and again, like that proteins have a hydrophobic part also, so it can interfere with the plastic of the syringe. It can interfere with the glass of the vial or the syringe. So we have to make sure that we have the right surfactants to avoid these interactions. When all this is being done, how do you test that you have the right formulation? So when we say that a formulation has to last for say 2 to 3 years at say 2 to 8 degrees, we cannot keep on testing every formulation and then come to know a result after 2 to 3 years. So we do stress testing, we do accelerated testing where we increase the temperature and then study the protein degradation in different formulation, and then choose the formulation which we find is the most stable under stress and higher temperature, higher pH conditions. So that helps in early screening of the formulation. But we have to generate data for the long term storage. So storage at 2 to 8 degrees for 4 years, 5 years also has to be studied. And during all these studies, which are short term or which are long term, like I said, all product quality attributes have to be monitored and make sure that the formulation is able to hold the protein with the quality that we have, we intend to. So once we have chosen the formulation, we go to the drug product process step. So after the downstream, we ended by saying that we can have a concentrated product which is highly pure, but the product is in some container. It can be in a bag, as we see in this diagram, or it can be in a vessel. And then what happens as part of drug product step, we have to add these excipients, which we have finalized during a formulation development step. So we have to add one by one all these excipients, filter it, and then we can store this protein under the formulated conditions. This now the protein with all these formulation is called as a drug substance, and we can store it for till we go into filling. So during all this step also, once we have known the formulation, we still have to, as part of process, we have to understand how do we add these excipients. These excipients have to be added either as a stock, should we make a concentrated stock, or should we add as powders. If we make a dilute stock, there will be a lot of dilution which will happen. What is the order of addition? The order of addition also plays a very important role. How long should you mix? Because these proteins are also shear sensitive. So what is the right amount of mixing? What test do you do when you're making these formulations, when you're making this product? And then for example, when we are going to the drug product, which filter to use, because proteins can bind to the filter. That is one. Or the other thing which can happen is some of these proteins or some of the excipients in the formulation can leach something out from the filter. So because the filter also is a chemical substance, so it can affect the product, plus the product can also affect the filter, and it can leach something out of the filter which can be dangerous for the product. So we have to study all that, whether to store in a bag or a vessel or a tank. What temperature should it be stored? Some of them are stored at -20, some at -40. So there are different stability at different temperatures. How do you freeze thaw? So that proteins are sensitive to freezing. If you don't freeze them right, the proteins can just fall apart, or after freezing, if we are thawing them, if we don't thaw them properly, again they can get degraded. So what is the freeze thaw cycle? How long can you store this frozen product? Everything has to be studied as part of making the drug product process. Now, so this is an example of the drug product process where we now from these frozen bags, we can, once we decide that now we have to fill the product into, a vial or an injection, then we take these bags, we can pool some bags, or we can have one bag to one drug product. It depends on the volume that we need. So once we make this drug product, it goes through series of filtration steps as final filtration steps, and then it goes to the filling machine where it gets filled into a vial, a cartridge, or an injection, which is like a syringe. So there also it is important to find out what testings we should do, how do we mix the protein, how do we what testing we do before filling, what testing we do after filling. Everything has to be developed as part of the drug product process. So going to the next slide, this is a busy slide, but what we want to show from here is that, I talked about developing the formulation, developing a process to be able to make the product, but another you know, is it, am I audible or am I muted?
The last few seconds,
Last
it was
few
not
seconds.
audible, Ankurji.
Okay. But should I continue or should I go back?
Only last for one sentence which you made, the comment. Only that if you can repeat and continue to be.
Okay, on the same slide?
Yeah, in the same slide.
Okay. So what I said is that in the previous slide, we discussed how do we make a formulation. Then we discussed how do we make a process to be able to, convert that formulation and the drug into a product. This slide is talking about what do we have to take care when we are choosing the final, we call it as a container closure. It can be a syringe, it can be a vial, the type of glass which is there, the type of stoppers, the type of capping, everything has to be studied because different proteins have different quality, different excipients have different qualities, and we have to make sure that we study everything and choose the best for every protein. On the right hand side, you can see some of the tests that we have to do. So like I said, product related tests are aggregates, oxidized impurities, because certain amino acids can easily get oxidized. Truncated species are fragment, so there are antibodies which can lose, say, one arm, which is like one a light chain. All these have to be studied. There can be some process related, like I said, some DNA coming from the cells, some protein coming from the cells. Everything has to be removed and tested to be proven that it is removed. Microbial safety is a very important thing, and that is why we have to make these products in a strict GMP environment, so that no artificial infection comes in the product. So and any endotoxin, any other bioburden, sterility, everything has to be checked. And like I said, there could be some extractable leachables which can come throughout this process, and we have to continuously monitor them, that they don't infect affect our product. So if I go to the next slide, so what we did till now is identify the product, use the machines, develop the process upstream, downstream, and formulation. So now we'll quickly go to the manufacturing of the product, and I'll just quickly cover this. So the concept is that all the work that I explained in the previous slides is developed in the lab or the pilot scale, and now we have to scale up to the manufacturing scale, where you can see all the columns become bigger, the bioreactors become bigger, and this is also a very, very important skill to be developed to convert a process from a small scale to be able to run it at a larger scale and run it consistently. So certain principles overall, if you see, and I'm giving an example of the upstream process right now. What we do is we develop and we optimize the process in the lab so that we get the desired output. The output, it will be in terms of say growth of the cells, the amount of protein that we are getting, the impurities that we are getting, the quality of the protein, everything is optimized, and we find the best environment, media, and the process condition to be able to get the quality and quantity that we want. As we scale up, what we try to do is, can we create the same environment which we created in a small scale to a larger scale? And the hope is, if we are able to replicate that, we will be able to preserve the quality that we got it at the small scale also at the large scale. So this is simplified objective of a scale up, that you can maintain the right conditions even at a larger scale so that your output is still the same. So just another example, how do we do that? So first we have to convert all the parameters into scale independent parameters, because the same values will not work between the scales. And then it gets divided into two parts. One is, for example, the type of the reactor in upstream, the bioreactor, and how do you operate the bioreactor, what air you give, what pressure you give. Now, the type of bioreactor gets fitted within facility. So, which means that for every process, we don't make a new bioreactor. So, once a bioreactor is there in a production facility, we have to try to make a process which will fit into that facility. And the same thing is also true for a downstream and a formulation, thing, that there are certain things that which are operational parameters which you can change from process to process, but you cannot keep on changing the whole unit operations for every process. So this is generally about the principles that we use for scaling up the process. Testing of the product. So like I said, during whole development and manufacturing, we have to continuously test the product and make sure that we are making it right. And not only during the manufacturing, but also during the shelf life of the product, we have to make sure that the quality of the product is retained. So certain tests, these are some example tests that we do. So on the first block, you see in process. So these are the typical tests which we do during the processing, when the product is getting made in upstream, downstream, formulation, which again I said aggregates, fragments, DNA, glycosylation, virus, pH, osmolarity. All these are the steps, and then after checking, the product moves to the next stage. Then DS is the drug substance. So once the drug substance is made, we have a battery of tests, after which we release the drug substance. And the release of the drug substance is to go to the drug product facility. And if you can see in the drug product side, we have a lot more tests, because this is the final product which is going out for the patient. So a lot of testing is part of the release for the drug product. And only once all these acceptance criteria are made, are passed, then the product is set to be released. On the other, you see the similar type of test that we have to study over the stability of the drug substance and the drug product. So most of these tests are also studied for long term, 2 years, 3 years, 4 years. It's different for different product, and we have to make sure that these qualities are retained. So this is the testing part. With this, we come to the last part, which is, the another most important part for a drug development is that getting, approval to be able to make sure that the drug can be given to the patient. So all this, development history that I explained, this is all part of CMC. So we have to compile all this data that we have generated over years of development work. This becomes a package, and then we have to combine all the clinical data which is parallelly running. Like I said in the first slide, different clinical trials are done. So all this package is put together, and then it is submitted for regulatory approval. Some examples I have given here. There are, for different countries, there are different regulatory agencies. Like we have in India, in US, in Europe, in Japan. ICH is International Conference of Harmonization, which tries to harmonize, different regulatory guidelines. WHO, and then each country has its own guideline, which is very specific to the need of the country. So when we develop a process, we have to make sure that because Biocon develops all process for global markets. So we have to make sure that when we are developing any process, we are meeting requirements from India, we are meeting requirements from USA, Brazil, Australia, Mexico, every country, because finally we want approval in every country. So like I said, approvals are based on dossier submission. Dossiers are the full package which goes for all the work which happens when we submit these packages to the regulatory agencies. They review the package, and if they have any clarification, they write back to us, and we are supposed to respond to that. So this is called query responses. And then physically also the regulators come and audit the facility. So if they have to find out whether the facility is good enough to be able to make good quality product. So when the regulators are satisfied with all the data, all the queries that we have responded, and the facility audit, that finally leads to the approval of the drug. And after the approval, we are allowed to market the drug in different parts of the world, depending on whichever country we have taken approval for. So I think in a very express way, I try to cover all the key areas for the drug development that we generally go through. And then if there are any questions, I'll be happy to answer if I can.
Can I come in? Mr. Ankur Bhatnagar, thank you very much. Absolute compliments. I really enjoyed your talk. Such a complex subject. Way you presented was so clear. Grateful to you. Thank you so much.
Thank you.
And
Thank you.
you have brought out all the nuances from beginning to end. I have one question. Given the regulatory framework in India and difficulties with regard to trials, testing, including facilities, the difficulty with animal trials, human trials, and all that. How do you feel, compared to other countries, how easy or difficult it is to kind of, bring the product to this level?
So it's difficult to say easy or difficult because every country has slightly different requirements, and which is based on the what that country actually needs. For example, animal testing, we have a requirement for animal testing as part of Indian approval. But what happens? Some of these drugs they don't have the right animal model. So in that case, we go to the agencies, and then our regulatory team, they discuss with the agencies that for this product, doing this animal testing in this model will not help because this is not the right animal model to be able to do that. So sometimes we get approval for that, and sometimes it's a discussion. And what also I think the good thing from the Indian regulatory side is that they are open to discussions from the companies also. So they have been taken views from the industries. So our senior people from our regulatory side, they have been called, they are heard. So I think this is appreciable that the Indian agencies make an effort to hear the other side and their challenges, and they also involve we some of our senior people have also got involved in regulatory guidelines, framing the regulatory guidelines. So I
So
think that is a right.
it is very participatory.
Yes, yes.
So, Mr. Bhatnagar, thank you so much. It was wonderful. Look forward to
Thank
meeting
you.
you sometime. Please do come to the academy
Sure.
sometime. Thank you very much.
My pleasure.
Thank you very much.
Sir, there is one question. How can we remove endotoxin? Can you please explain on this?
So endotoxin generally so there are different ways. First of all, we prevent the entering of the endotoxin. That prevention is the key. So for example, starting from our water, the testing of the water is done. We use water for injection. We make our own water. We have the water plant. So the water is tested because water is the key. Media is prepared in water. Buffers are prepared in water. So we test the media and buffer and make sure that they are endotoxin free. Any endotoxin that can come from anywhere in the process, we have these chromatography steps like the anion exchange chromatography. It takes care of the endotoxin, and further in the process, like I said, we keep on checking the endotoxin levels and make sure that they are controlled. But to answer that question, there are two things. One is try to prevent the endotoxin coming in, and the second is the anion exchange chromatography step, which takes care of endotoxin.
Thank you very much, sir. There is one more question from Milin. What kind of biological drugs are currently manufactured in Biocon Biologics?
So we have all type, like in the beginning slide, I said we can use bacteria, we can use yeast, and we can use mammalian cells. So we use all three in Biocon. We use bacteria to make some product which is there. We can see on our website. Pegfilgrastim recently got approved in USA. Then we use Pichia to make all insulins and analogs. So insulin, glargine, Aspart, all these are insulins and analogs of insulin. We use Pichia, and then we make monoclonal antibodies using mammalian cells. So there are I think three antibodies from Biocon which are in market, and one or two are under the approval stage.
Thank you very much, sir. There is one more question. Why measure the higher order structure of the proteins?
The question is, why do we measure?
Yes, sir, exactly. Why do we measure
Yes,
the
yes.
higher order?
So we have to measure because it impacts the stability of the protein. If you have not made the right protein in the beginning, it can behave slightly differently. So higher order actually helps us to say that from the starting, the primary is from the sequence to the actual folded protein is all made in the right way. And sometimes some of the drugs that I didn't mention about that, but we also work on biosimilar drugs, which are, so there is an innovator, say antibody, and we are trying to make an antibody similar to the innovator. So there, having the right higher order structure gives the confidence that our antibody will behave very similar to the antibody which is there from the innovator. So that is why it's important.
Thank you very much, sir. There is one more question from Mahi Priya. Does Biocon works on synthetic chemical drugs also, or it deals with only biological synthesis?
No, we do chemical also, and the Biocon does a lot of these small molecules. It's a different division where we do small molecules. Both we make small molecules through chemical synthesis also, and also through fermentation route. So we do. it's a big business for Biocon.
Thank you, sir. What kind of in silico studies you perform for identifying the product needed?
So in silico studies, we generally try to get different cell lines. So, for example, if we are making a drug which is supposed to bind certain target, then we get a cell line or make a cell line which is expressing that target, and then we as we are making that drug, we try to study the binding of that our antibody to this cell line. We also we have to study the binding and the release. So if there's an absorption and desorption. So all that kinetics have to be studied. If we're making insulin, then we have to use cell lines which are sensitive to the glucose uptake, and we use them to understand the insulin that we are making is working in the right way or not.
Thank you very much, sir. There is one more question from participant. Which safety level is maintained during the process?
So generally here, the safety level that we have, because we don't work with the virus, so our safeties are all for the product, not for we don't have to safeguard for the virus. Our facilities are completely virus free. So generally clean environment which is there, which is so all the sterile operations we do under the laminar flow, the normal laminar flow, and our manufacturing plants, they are all have these HEPA filters facility, which is under classified as class 10,000 facilities, and that is what we maintain. If there is a sterile operation, that happens under the class 100 in the laminar flow type environment.
Thank you very much, sir. What about pre-formulation studies?
Formulation studies are yes. So generally, before we study the formulation, we try different like I said, excipients and different conditions. We have to find out, like I mentioned, what is the right pH for our formulation, what is the right stabilizers for our formulation. So we study these. We study the nature of the protein. We have to study the glass transition temperature, isoelectric point of that protein. So all these and different pre-formulations we do to narrow down the formulation that we want to now pursue. A lot of pre-formulation studies.
Thank you, sir. There is one more question from Vipin. If seen any change in the protein secondary structure, will it impact the protein stability, aggregation, quantitation?
Yes. So
And
it yeah.
similarity.
So it will impact, and like I said that the protein actually goes through different steps during the process. So once it is made in the upstream, it goes through different purification steps, formulation steps. So if the protein structure is not right from the beginning, when the different steps have different conditions of conductivity, pH, temperature, other components. So there is a high likelihood that the protein will not be stable.
Thank you very much, sir. Now all the questions and the answer sections, we have answered most of the questions. We will take offline questions and send you also through mail.
Sure.
Now
Yeah.
we will come to the last part of this webinar. Sir.
Dr. Ankur Bhatnagar, it was a very interesting and insightful talk. As our chairman sir told, we actually broadened the horizon of our knowledge in drug development. As we know, it's a very complex thing. You made it very simpler. Thank you very much. We will be requesting you to give more such talks in our